Hey guys, so today in this lesson, we're just going to be going over the first 10 slides of the male reproductive unit. Back when schools got closed because of COVID-19, we had just done a little bit of the male reproductive system, and then we're planning on finishing it the next day, and then the next day schools were closed. And so I already have the second part of this lesson recorded, so I'm just going to be recording first part in this video. So anyway, let's get into it. So the male reproductive system, for this we're going to be going over the different structures of the male reproductive system, but also some of the specialized cells that make up the male reproductive system and help it function. We're going to go over how sperm are formed and the difference between sperm and semen, and then some of the different functions of those different structures and specialized cells. So sex characteristics are divided into both primary and secondary sex characteristics. The primary sex characteristics are the actual structures that have a direct role in reproduction. So things like the testes and the penis in males and ovaries and the vagina in females. And these sex characteristics develop around the 3rd month of embryonic development when the sex chromosome genes cause differentiation between males and females. And then these secondary sex characteristics are structures that are not directly related to reproduction, but are distinct between males and females. Those include things like body and facial hair, muscle development in men, breast development in women, things that tend to pop up around puberty. For the male reproductive system, we're going to be looking at all of these different structures: the scrotum, testes, seminiferous tubules, Sertoli and interstitial cells, epididymis, vas deferens, Cowper's glands, seminal vesicles, prostate gland, ejaculatory duct, urethra, and penis. So for each one of these things that are on this list, you need to know where they are, what they look like, and what they do. This is our diagram of the male reproductive system. Pretty much everything that is on here you should be able to identify, though you don't have to be able to pick out the foreskin or the anus on a diagram. And the ureter and the urinary bladder are really just useful to identify so that you don't get them confused with some of the other structures that are on here. But essentially, as we start going through the male reproductive system, we're going to be talking about the testes, which is where sperm is formed. Then we'll go up to the epididymis, where sperm is stored while they mature. After the sperm leave the epididymis, they travel through the vas deferens, which is this long tube here. They then pass the seminal vesicles. There are multiple of them, but we can only see one from this angle. There would be another kind of on the other side of the urinary bladder. It is important to note that the sperm does not go through the seminal vesicle. It just passes by the seminal vesicle, and the fluid in the seminal vesicle is added to the sperm. The sperm then continues to travel, going again just past the prostate gland. Once again, the fluid from the prostate gland will join the sperm, but the sperm does not go directly through the prostate gland. And then same with the Cowper's gland, which is this little yellow spot inside this pancakey bit here. Again, the fluid from the Cowper's gland will intermingle with the sperm, but the sperm does not go through the Cowper's gland itself. And then you'll get into the urethra, which is the tube that then carries the semen through the penis and out. The scrotum is the pouch of skin located below the pelvis. During the last 2 months of fetal development, the testes will descend out of the abdominal cavity and into the scrotum. And then a membrane forms over that canal that the testes descended into so that they don't, when you do a handstand, go back up into your abdomen. Since the scrotum is away from the body, it is a few degrees cooler than body temperature, which is ideal for sperm production. If sperm get too hot, they will die and won't be able to develop. So it is important that they are kept below body temperature. The cremaster muscle is the muscle of the scrotum that controls the temperature of the testes. Essentially, if the sperm are getting too cold, that muscle is going to contract and bring the scrotum and the testes closer to the body to warm them up. If the sperm are getting too warm, which is affecting their production, the cremaster muscle will relax so that the testes descend further away from the body so that they can cool down. The testes, aka testicles, are the two male gonads responsible for the production of both sperm and testosterone. Inside of each testes, you can see, is divided up into these different lobules, which are these little kind of flower petally parts. And then inside each of those lobes are these seminiferous tubules, which are the noodly bits that you see inside here. Inside these seminiferous tubules is where spermatogenesis or sperm formation happens. The inside of these seminiferous tubules are lined with spermatogonia, which are these sperm-producing cells. Each spermatogonia contains 46 chromosomes. During spermatogenesis, which is again the way that we produce sperm, the spermatogonia are going to divide into spermatocytes, which then differentiate into 4 spermatids. That process is meiosis, and during meiosis, if you remember from I think like grade 9 of biology, during meiosis the number of chromosomes is divided in half. So even though the spermatogonia have 46 chromosomes, these spermatids are only going to have 23 chromosomes. Those spermatids are then going to develop into the sperm cells. So this kind of describes that process. This kind of dark orange layer here is the outside or the membrane of the I have the word spermatogonia stuck in my head, seminiferous tubules. And that would form like a full circle if we went the whole way around. This is like kind of like a cross section of the seminiferous tubules. So essentially we have the spermatogonia on the outside. Those are 2N, which just means they have 2 copies of every chromosome, so 46 chromosomes. That spermatogonia is going to divide using mitosis to form 2 more diploid, or cells that have 46 chromosomes. One of those, which is daughter cell type A, that one is just going to stay a spermatogonia. It can be used later on for more spermatogenesis. The daughter cell type B is the one that's actually going to undergo meiosis to become the sperm. So that daughter cell type B spermatogonia is going to become the primary spermatocyte. It will divide using meiosis to become the secondary spermatocytes, and they are now haploid, meaning that they have half the number of chromosomes, so they only have 23 chromosomes each. Each of those are then going to divide into these spermatids, so we end up with 4 spermatids. Each one of those will develop into a sperm. Okay, and then this is where we pick up in the second part of this recording, so that is all that we're going to have in this video.